you should pitch that in the end. You're looking for postdocs.
Cool. I'll verbally mention it.
Yeah.
You used to have those
sorry, you were saying
quizzes,
something?
no? You used to have those quizzes, no? At the beginning.
Yeah, Jen used to
stuff.
do that.
Yeah.
Right, Jen, in the beginning when you guys started, you used to have those quiz kind of a thing where you would put like the favorite words and stuff like that.
Yes. Yeah, we did a cat versus dog people, and I was very sad to see that dogs won. I was like, what's wrong with cell migration people?
I remember that one. I didn't vote because I'm not sure which person I have. I have none.
We did collective migration versus single cell migration, which is your favorite, and that was like a 50-50 split, which was interesting.
Yeah.
But yeah, that was all Becky. She was a grad student along with Adam and Roberta Mayer's group, and she has moved off to bigger and better things. So we're not fun anymore because neither you, Agata, or I have the bandwidth to be fun.
I'm an end of my postdoc. This is all the fun I can have right now.
Exactly. Maybe one day when the most fun is being about to graduate. That's right. Being about to defend.
I do feel that it's an exciting time, right? Like once you get your, it does feel like a major accomplishment. At least it did to me because it's literally blood, sweat, everything.
Yeah, I would say it is a major accomplishment. Doesn't it just feel like it, right? Yeah.
It's literally the highest degree you can have. And
right. So yeah, I think we're all set with the tech stuff. So
perfect. Yeah. I think we are kind of on time. Okay. While people trickle in, I will start introducing Purnati. So welcome everyone for another session of Cell Migration Seminar. Today we have two wonderful speakers, Purnati and Nils. We will go with Purnati first. Purnati is a microbiology student from University of Delhi in India, and currently she is in Tamal Das lab where she's trying to solve a decade-old puzzle of how cells orient the Golgi apparatus position during migration. In the last few years, she has discovered a previously unknown cell migration-induced Golgi remodeling process, which I think she is going to be talking about. When Purnati is not in the lab, you're likely to see her cycling to a volleyball court, which I find exciting because I think I do no sports right now. Welcome so much, Purnati, and we're really excited to have you here today. I also quickly wanted to mention, which I think Purnati is going to say, but she is going to graduate soon and she's going to be looking for postdocs. So Purnati,
shout out.
this place is yours now.
All right. Okay. Welcome, everyone. I would like to begin by thanking the organizers for this fantastic opportunity and the platform. So today I'm going to talk about emerging state of Golgi organization and collectively migrating epithelial cells. So Golgi apparatus, since its discovery, has been very frequently used to define cell polarity and how the polarity within the cell changes. Here, there are many examples. I'm just showing you a couple of examples here during the corneal epithelium development in chick embryos, where the apicobasal Golgi position completely switches to a basal position in the cells. And more recently, people have been using it to define cell polarity in experimental wound healing conditions, where Golgi orientation aligns towards a direction in cell migration. And people have very frequently, this is where Golgi polarity has been used. But if you think about it, the Golgi polarity is very crucial to define the cell polarity within the epithelial cells, where the epithelial cells try to or tend to organize their intracellular components like nucleus towards the basal surface of the cell, and the Golgi and other organelle like centrosomes is towards the top of the cell, defining the epicobasal polarity in epithelial cells. But this is in epithelium when it is stationary, it's not moving. But when under certain conditions, let's say wound healing, epithelial cells start to migrate, and the Golgi position from the epicobasal position switches to a position in front of the nucleus, somewhere in the XY plane. So this change in Golgi polarity has been known for a pretty long time, but what we do not know is how this change or how the position of Golgi changes in a 3D space of the cell. So in the next few slides, I'll try to show you how the Golgi position change, and we'll try to see we'll try to understand how it affects the Golgi organization, as well as how the molecular players what are involved, the other molecular players that are involved in this process. So to look at basically to answer the first question, how it happens, we started looking at migrating epithelial monolayer. We have used for most of my talk, I'll show you data from MDCK cell line, but we have also verified in this KQ2 epithelial cell line. So we first grow an epithelial monolayer by culturing them inside a culture insert, and we lift the insert to allow the cells to migrate, and we let the cells migrate for a few hours, and then try to look at what are the different kinds of organization Golgi can take. So if you look at this panel here, I'll switch to laser pointer. Yeah. So if you look at this panel here, we are looking at a cell that has not yet started migration, and we can see the Golgi marked in green here stays towards the top surface of the nucleus, marking the apicobasal Golgi polarity. And when we look at cell that has already migrated for a few hours, the Golgi comes in front of the nucleus, defining the front rear polarity. But we also found one intermediate state where Golgi disperses around the nucleus in a perinuclear fashion and forms a ring-like organization around the nucleus. So for the time being, we are calling it equatorial Golgi position. And we found that this is a time migration and time dependent process. And as the cells, more and more cells migrate into the wound, the number of cells with the equatorial Golgi marked in green here increases with time. And as soon as it has gone through the intermediate state, this Golgi attains a polarized state, and you see an increase in number of polarized cells at the edge of the migrating monolayer. So this is not just a marker specific organization we see this equatorial Golgi organization using different markers for different cisternates of Golgi. And when we also try to follow it with live cell imaging using a cell line which expresses Golgi, fluorophore targeted to Golgi all the time. So again, we also recapitulate whatever we find in our fixed cell imaging. And if you try to quantify the Golgi vesicles and how they are moving in time with respect to the nuclear centroid, you will notice the Golgi starts with most of the Golgi vesicle stays on top of the nucleus at the beginning of the migration. Towards let's say after 2 hour or 2.5 hour of migration, it enters the intermediate equatorial dispersion state, and it stays in the nuclear plane after it attains the front rear polarity. So what we found here was a migration dependent Golgi remodeling process. So we wanted to see if it has anything in common with an already existing Golgi remodeling process, the mitotic Golgi remodeling. During mitosis, to ensure equal partitioning of the Golgi element within the two daughter cells, the Golgi undergoes a complete fragmentation where the ribbon-like organization disintegrates completely into small vesicles, and very frequently it is visualized by using a marker protein called GRAS65. And you can see here as the cell proceeds through different states of mitosis, the GRAS65 undergoes gradual fragmentation until all the signal becomes a haze. So we try to use the same marker to compare to see if our remodeling we see during migration has anything in common with mitosis. And as you can see here, in mitosis, Golgi undergoes fragmentation. We see loss of GRAS65 signal. But in case of our transition state, Golgi remains in a uniquely perinuclear organized state, and the GRAS65 signal is again intact, tubular, as you would see in a non-dividing cell. So with this and several other experiments that I'm not showing here right now, we were able to verify that this is different from mitosis, and Golgi does not go any kind of fragmentation and maintains a ribbon-like organization throughout this process. So with that, we were able to kind of conclusively say that we have discovered a migration-induced Golgi apparatus remodeling, where Golgi goes through an intermediate step and undergoes a dispersion in a perinuclear fashion before getting polarized in a specific direction. So to see how to understand how physically one cell can move around the Golgi in a way that it takes an equatorial shape, we try to look at the physical mechanisms that can allow that the physical movement of Golgi or any other organelle within the cell. So the first candidate was obviously microtubule. And when we tried the classical approach of disrupting the microtubule and trying to see the effect of it on Golgi organization here, as you can see here, Golgi undergoes a complete fragmentation. So if this experiment kind of does not help us to address if microtubule is involved here or not, so we had to devise an approach where we can keep the Golgi structure intact, the microtubule intact, and still be able to comment if the microtubule is involved here or not. So to do that, we looked for, microtubule binds to interacts with Golgi through some linker proteins. We looked for some linker proteins and created mutants for try to create mutants for those linkers, so such that the Golgi does not bind to the microtubule filaments anymore. And we found one such mutant, one such protein called AKB450. We created a mutant, we got a mutant of this protein and tried to see what happens to the mutant cell and the Golgi organization in these cells. So here we are looking at again at a migrated epithelium, and the mutant cells here are marked in magenta, and you can see here even the mutant cell where the microtubule has been dissociated from Golgi, the cells are still able to go through the perinuclear Golgi dispersion just like wild type cells. And if you quantify all the 3 different types of Golgi organization that we have seen so far, there is no significant change in case of the mutant. We also looked at how the microtubule is organized in control and the mutant cells. In control cells, as you can see, the microtubule nicely starts from Golgi, both in the polarized state as well as the equatorial state, and it forms kind of a nice ring around the nucleus in control cells. But in mutant cells, as you can see here, the microtubule has is vanished from the Golgi area. It does not start from Golgi anymore. So, and you also see loss of this perinuclear microtubule arrangement. Okay. So this kind of showed that the Golgi associated microtubules are not involved in this migration-induced Golgi remodeling. So we looked at the other cytoskeleton that has been widely reported to be involved in maintaining Golgi position within the cell. So we look that is actin, and we looked at actin and how it could be involved in this process. First, we again, just like microtubule, we started with some drugs that can perturb actin dynamics within the cell. What we found was either if you stabilize the actin filaments or disrupt the actin filaments, in any kind of perturbation to the actin dynamics leads to loss of equatorial Golgi in the migrating epithelium or the cells. So when we quantify the number of cells with all the different kinds of distribution, in case of these drug treated cells, you do not see any cell which has equatorial Golgi. So that was a huge clue that for us that actin is probably involved here. So to study that further, we switched to live cell imaging with actin and Golgi together. And what we found was this interesting actin ring-like structures, which starts which starts with the small actin filament coming out of this cortical actin and forming ring around Golgi vesicles like this. I hope this is clear. So how this kind of Golgi actin interaction could be affecting Golgi organization state. So to understand that, we did few more live cell imaging, and what we found was this kind of actin interaction can lead to 2 different kinds of effect on Golgi behavior. Either it can lead to the ribbon disentanglement, where it basically cleaves off a network-like point and allows the ribbon to be more longer, or it can be like in the form of where the actin ring slides over the Golgi ribbon and fragments the Golgi ribbon. So both of these, this kind of interaction can basically disentangle the Golgi network sitting on top of the nucleus, and it allows the dispersion, loosens of the Golgi network to allow dispersion around the nucleus. So to understand what is the nature of these actin projections or actin rings that is coming out of the cortical actin, we try to look at what are the different actin nucleation factors that could be involved here. And what we found was if we inhibit R2/3 mediated actin branching, it leads to the loss of this actin projection within the cell. So here we are looking at a time projected image, and all these different color dots or puncta you see are the dynamic actin, the actin projection coming from the cortical actin. In absence of drug, in absence of the R2/3 inhibitor, you see many actin projection within the cell. When we add the drug, you see a significant decrease in the number of actin projection, and when we again remove the drug after washout, we see a recovery in the number of actin projection within the cell. So this is what happens to actin during if you treat during this process, and what happens to Golgi if you let the cells migrate in presence of this drug. So in control cells, as you can see, we see many cells with equatorial Golgi dispersion, but in R2/3 in presence of R2/3 inhibitor, where we do not allow R2/3 mediated actin branching, so cells do not go through any kind of equatorial Golgi dispersion, and that is reflected in the quantification as well. So what we found was there is an R2/3 mediated actin branching event that is leading to formation of smaller actin rings, and that is leading to Golgi remodeling by either Golgi ribbon disentanglement or Golgi ribbon fragmentation. But what we asked then was how is this being mediated at a protein interaction level, and how the actin dynamics is being translated into Golgi dynamics. So to do that, we also followed a similar approach like in microtubule. We looked for linker proteins that can mediate interaction between the actin filament and the Golgi apparatus, and create mutants for these linker proteins to dissociate the actin filaments and the Golgi apparatus, and see what happens to equatorial Golgi dispersion in these mutants. So what the literature tells us that the Golgi can bind to actin through two different molecular axes, the GRAS65 MENA pathway or the Golf3 myotin A pathway. GRAS65 and Golf3 reside on Golgi membrane, and MENA and myo18A bind to actin directly, and the Golgi and actin interaction is mediated through these proteins. So before going for the mutants, we first looked at the endogenous localization of these proteins within the cell. For Golf3 and myo18, as you can see here, there is localization to Golgi, but there is also significant non-Golgi localization within the cell. And for MENA, what we found was MENA specifically localized to Golgi only in this case. So to create mutants, what we did was for both of these different axes, we created different mutants. For the Golf3 myo18A axis, we created a myo18A mutant by overexpressing the Golgi binding domain of the myo18A within the cell. And for MENA, we created few mutants. MENA can bind to Golgi through EVH1 domain or actin through EVH2 domain, and it can bind to globular actin through GAP domain or the filamentous actin through the FAB domain. So we created deletion mutants of both GAP and FAB domain, and with all these three mutants, try to see what happens to Golgi, the equatorial Golgi dispersion in migrating cells. So if you look at here and follow with me, in case of the myo18 mutant, we are expressing the Golgi binding domain of this protein. The mutant cells marked in magenta here again go through the equatorial Golgi dispersion. The deletion mutant for GAB also goes through the normal equatorial Golgi dispersion, but the deletion mutant for the FAB domain of MENA protein, and in those cells are not going through the dispersion, the equatorial dispersion. Rather, they are polarizing, achieving the front-rear polarity directly. So when we quantify the percentage of cells with the equatorial Golgi dispersion, in case of the MENA FAB deletion mutant, we see a significant reduction in number of cells with the equatorial Golgi position. So, but there is a catch here. So MENA, although in our case we see a very specific localization to Golgi, it has also been reported to localize to focal adhesion as well as cell-cell junction through the FAB domain, the filamentous actin binding domain. So to avoid any effect that we may see from defect in the cell migration itself, we created another mutant for this MENA protein by just overexpressing the EVH1 domain of this protein, the Golgi binding domain of this protein. And in this case, again, what we see is the wild type here. goes through the normal equatorial Golgi dispersion. The mutant marked in magenta here, the Golgi stays apical on top of the nucleus at this point of migration. Eventually, if you let the cell migrate, they become front-rear polarized without going through the equatorial Golgi dispersion. So, but the uninduced, so this entire EVH1 cassette we put it under a inducible system. So only if you induce it, this the mutant effect, the phenotype of the mutant we see, and in uninduced control, the cells proceed through the dispersion state normally. So so from this, what we can say is the out of the two molecular pathways, probably the GRAS65 MENA pathway is involved. So to verify that further, we also try to perturb the GRAS65 MENA interaction by creating a truncated GRAS65 protein. So briefly, GRAS65 has two domains: the GRAS domain required for the oligomerization of the protein, and the SPR domain, which gets phosphorylated during mitosis to allow fragmentation. So, but the MENA binding region resides in this SPR domain. What we did was we created a truncation mutant of this protein, where we only have the GRAS domain. So, to get rid of any effect that we might be inhibiting cell division in these mutants, we again put this entire construct under a inducible promoter system. And when we induce the expression of the mutant, again, as you can see here, the mutant cell does not go through the equatorial Golgi dispersion, but the wild type cells do. And the uninduced control also goes through shows a similar behavior as wild type, and which is reflected in the quantification again. Since in this particular mutant, we are inhibiting GRAS65 and MENA interaction, there should not be any MENA localizing to Golgi, and we verified that. Here you are looking at the mutant cell marked in magenta here, and this is the staining for MENA. In the control cells, all the other control cells, you see it's localizing to Golgi nicely, but in the mutant cell, you do not see any Golgi specific MENA localization.
Purnati, sorry to interrupt you, but you might have to wrap up because we're 20 minutes in.
I just have one more slide after this. Sure. So from this we found out that the actin dynamics and Golgi dynamics are mediated through the GRAS65 MENA pathway. Now coming to why is cell taking so much effort and going through this entire process? Why cannot the cell just switch to a frontier position directly? So we compared what happens to cell migration behaviors in control cells, which are going through the equatorial distribution properly, and the mutant cells which do not go through the equatorial dispersion. And here we're looking at the migration trajectories of different cells, and the direction of migration is towards this. The wound would be somewhere here. And all the control cells show a directional migration towards the wound. But in both the mutants we see a loss of persistent directional migration, and they just migrate randomly around their position. We also looked further at the migration trajectory and looked at the angular changes at each step. For control cell, there is very less change in angle at each step. But for both the mutants, what we see is a very high degree of changes at each step, high degree of change in theta at each step, which again results in the net displacement in these mutant cells being very less compared to the control cells. And finally, we also found, other than this defect in cell migration behaviors, we also found there is a fault in intracellular reorientation of other cellular components as well. So here, in this case, we looked at position of the centrosome using gamma tubulin, and what we see is there is an increased distance between Golgi and the centrosome, and it also shows a more variation in the distance between Golgi and centrosome. So put together, what we have found is there is a migration-induced Golgi remodeling process that is happening in epithelial cells. And during this remodeling, Golgi takes up an equatorial dispersion, and before getting polarized in a front-rear manner. And if this dispersion is inhibited, cells lose their directional migration ability. So this is our work, one of the work which shows the importance of perinuclear Golgi organization and the importance of it. I would just like to mention one more work where people have reported existence of perinuclear Golgi, but during development where myoblast cells or the muscle cells fuse to form the myotube. In that case also you see perinuclear Golgi. And this Golgi organization further gets stabilized when they form the neuromuscular junction. So put together, what these work are kind of suggesting towards an idea where the perinuclear Golgi has some functional importance, which only become apparent when you look at higher order, more complex systems like multicellular systems or tissue level development. So with that, I would like to thank all my lab members, especially Simran, for the expansion microscopy images, which I could not show, but still, and all the beautiful discussions we have with our collaborators, and again, the organizers of this event. Thank you.
Thank you, Purnati. We really appreciate that great talk. We actually are running short on time, so I'm going to ask anyone to put questions for you in the chat, and you can answer there. And with that, I will go ahead and introduce our next speaker. So we're delighted to have Nils Gauthier today. He started his training in 2000 at the University of Nice in France for his PhD, where he showed how H. pylori toxin hijacks the endocytosis pathways of their target epithelial cells. And then for his postdoc in 2005, Nils joined the laboratory of Professor Michael Sheets at Columbia University, where he showed how membrane tension orchestrates cell shape changes. He then went on to the Mechanobiology Institute in Singapore, where he started his own group, and they worked on multiple lines of research relating to membrane mechanics, including phagocytosis, membrane trafficking, and cytoskeletal remodeling. And they also worked on the development of microfabricated tools to mimic in vivo fibrillar environments. And then in 2016, Nils joined the IFOM in Milan in Italy and started an interdisciplinary mechano-oncology group studying the biophysical properties of tumor progression. And I think we'll hear about some of that work today. So we have an exceptionally international scholar with us, and we're really excited to hear about your work. And so with that, Nils, the floor is yours. Thanks so much.
Thank you. Thank you very much for this kind introduction, and good morning on the East, good afternoon on the middle, and good evening in Asia, as I have friends all over these places. Then today I'm going to talk about adaptive mechanoproperties of the formin FNN1, basically, like that is linked to the glioblastoma fitness for invasion. Then this is part of what we have done, the mechano-oncology unit. And the mechano-oncology unit, that's the lab we have in Milan, is deciphering the role of mechanical signal generated at the interface between membrane and cytoskeleton during tumor-associated biological event. Then we are working on 3 compartments, the supporting tissue with fibroblast matrix interaction. We have published many contributions on that. On the immune function with the study of macrophage biomechanics. And today, what I'm going to talk to you is about the third part, where we are looking really at the cancer compartment in this tumor ecology, with the brain cancer progression, with the study of glioblastoma invasive property. And this is really the work of Pascal in the lab, that is a scientist, and my wife also, and we work together since 20 years. Then that's a long story. Then, what is GBM? GBM, glioblastoma multiforme, is a very nasty tumor. The median survival is 14 months. Then GBM presents unique treatment. And if you look at the American Association for Neurological Surgeons, that really list what are the treatment challenge. They are localization, whereas the tumor in the brain, the resistance to conventional therapy, the limited capacity of the brain to repair itself, the variability of the disrupted tumor blood supply, because the cells are destroying basically the brain, and that impaired drug delivery. You have a lot of capillary leakage, edemia, intracranial pressure that leads to further damage to the brain, tumor-induced seizure, and the resultant neurotoxicity of treatment directed to glioblastoma. And among those, there is one that I hide purposely. And this is migration of malignant cell into adjacent brain tissue, which makes the purpose of this talk why we are focusing on this migrating glioblastoma. Then GBM, when they migrate in the brain, they preferentially use brain blood vessel as tracks, as highway. Then if you look at the primary tumor, they move along those blood vessels. And the treatment is basically to debunk this primary tumor, then to treat the border with radiotherapy, and then they give tomozolomide to try to kill the last cells. Unfortunately, you have many of those cells that were hidden and small and discrete that migrate, and then they can cause recurrence further on later on. Then this is really the goal of our work, is to identify those rare and those nasty migrating cells. Then the lab that really like showed the first time into in vivo slide was Peter Cannell in 2006, where they transplanted glioblast cell and they showed that they migrate and proliferate on host brain vasculature. It was a rat system here where there are rat C6 on brain blood vessel. How we can mimic that? Then the idea is to use blood vessel like mimicry pathway proxy. We use like micropentode device that here in that case are coated with laminin, because laminin is the proper substrate for migration of glioblastoma and not fibronectin nor collagen. And we have different grades of size that mimic capillaries or big arteries. Then we drop the cell on them. And what we have seen previously is that mechanical constraint mediated by the topology or the cell density triggered linear migration and really trigger, sorry, I have my cell phone. Okay, perfect. Sorry for that. Trigger the linear migration of the cell along this anti-parallel stream along the lines. Then we recapitulate that recently in a book chapter, if you are interested, in vitro mechanobiology of glioma, mimicking the brain blood vessel and white matter tract invasion path. And we have many techniques among those microfabrication, including the microcontact printing. Then what are the molecular players of this? Then obviously cell motility relies on the actin cytoskeleton, and the best players are R2-3 complexes and the formin complexes that form respectively the lamellipodia branch network or the linear like stress fiber tap network. For glioblastoma, and particularly, we observe that the formin FOD3 that localize at the tip of actin stress fiber near the adhesion site, and when you transfect this formin, you see that the cell increase filament bundling and increase linear filament. And this leads an advantage to the cell, because they can now migrate very well on linear tracks. To cut a long story short, what we show in that is that glioma laminin linear migration depend on this formin FHOD3. And if you compare 2D, where you have R2-3 dependent migration and formin dependent migration, and part 2D, particularly MDR1, it's not the case in 1D migration. It's not R2-3, it's not MDR1, it's some formins, and particularly FOD3, that emerge on this linear migration and that are relevant really to the brain environment. Then this shows you that topological and mechanical constraints dictate molecular switches. How we move that forward? Then we had a rat model. Then the idea was to go for a more patient-driven assay. Then we take this opportunity to use GBM stem cells. Then GBM stem cells are patient-derived cells that are isolated and cloned as tumor sphere. They are re-injected in a mouse brain, and if the mouse is generating a tumor, they are classified as an avatar, a proxy of the patient. Then we use those cells and we choose the 3 stereotypical behavior from 3 different patients. We had NNI11 that forms circumscribed tumor, hemorrhagic fast growing. We are on the other hand another patient that had NNI24, which are diffusive tumor, non-hemorrhagic, slow growing and slow killing. And in the middle, we had this super nasty tumor, NNI21 from this patient, which were diffusive, hemorrhagic, fast growing and fast killing. This was the edge of the study to define this extremely heterogeneous tumor. What we observe, we derive an assay how to combine linearity and capacity to mimic blood vessel, meaning turning. Then we develop a grid assay, and dropping the cell on the grid showed dramatic difference between those 3 cell lines. Then NI11 non-moving, NI21 moving very actively and jumping also from line to lines, and NI24 not moving by jumping, but really following the tracks, following the linear tracks on this assay. This allows us with a simple projection of the movie, a Z-stack projection, to determine what type of motility you have. This is the value of the assay, is that you can just look at the gaps in an NI11 between the cell groups and the gaps that show that it's not moving. You can see an NI21 are covering the non-paternad area, which show that they are jumping from adhesive side to other adhesive side. And the NI24 fill up completely the grid without filling up the square zone, the non-adhesive zone, showing that they are following the tracks. Then we have different protocols for that. Then you can find that in this recent publication also, where we basically delineated the substrate preparation, how to maintain those cells, how to transfect and express protein in those like glioblastoma, and also all the image tracking software related to the analysis on the single cell motility on grids. The second question, what are the mechanoproperties of this human glioblastoma cell in terms of cell traction force, cell adhesion, and cell stiffness? What we observe using traction force microscopy derived from laminin lines printed on deformable substrate is that the non-motile cells were developing very low level of force. But motile cells, whereas they were following the lines or jumping around, they were developing high force, but we could not differentiate the two types. Then we move on to analyze the cell adhesions. In that case, again, the two motile cells presented large adhesions, but the non-motile cell had no adhesions. But again, comparing the 21 and the 24 didn't highlight very well what was the difference to explain while one was very invasive and jumping around, while the other one was just following the tracks. Then we look at cell stiffness, and there something emerged. On 2D, the cell stiffness showed that the most motile one was the stiffer, but there was no major difference between the two other lines. And what emerged when you put them on grid is that the most motile one was still the stiffer, but the motile one that was following the grid was getting very soft on the grids. And this highlights a major difference that we had finally between the two motile modes. Then what are the molecular determinants of those differences? Then we look at the formins, because we knew that ARP23 was not very important. And among the formins, we compare spheroid versus spread cell on laminin. And what we observe is that there was a bunch of formin expressed for sure. We confirm FOD3 as one that was highly expressed in motile cell and non-motile cells, sorry. And one emerged, FMN1. FMN1 was only expressed in the most motile jumping cell, and was also induced by laminin substrate. Then we start to dig in. If we give those response of laminin, indeed, we had higher expression of laminin of FMN1, and this was specific to laminin. If you give more fibronectin, the cell don't increase the expression of FMN1. If you give more collagen, they don't also express FMN1 at higher level. Then we quantify that, and we looked what was the possible players. One of the obvious players was the YAPTAZ pathway. But the YAPTAZ pathway, while it was induced non-specifically on laminin and fibronectin, was not increasing the mRNA level of FMN1, while it was spreading well that all the targets that are known, among all the targets that are known to be under YAPTAZ pathway, they were induced by both substrate. Then it shows that we had a post-transcriptional regulation expression of FMN1, and by mechanochemical coupling with laminin. Indeed, laminin given soluble was not inducing FMN1. Laminin must be attached on the substrate. The next question, how those molecular determinants are reflected in vivo? Is it relevant for the disease? Then the answer is yes. What we had is two essay. One was xenograph. This is a bank that was available, and we interrogate this bank, and we observed that there was a higher level of expression of FMN1 in the area of lesion versus the tumor core. And we use also patient samples where we stain for FMN1, and we look at the core of the tumor or at the edge of the tumor, and we observe that at the edge of the tumor, there was more FMN1. Is FMN1 involved in this cell motility that is peculiar of the NNI21? Then for that, we turn to the assay and we look at 2D, and on 2D, we didn't observe much. And that was the beautiful approach that we developed, the grid approach, that really highlight the problems. And what happened is that when you remove FMN1, NNI21 cell, the most motile cell, were stuck at the corner of the grid, and they were not able to make decision. This has consequence that on the grid, the speed of the cell was dramatically reduced. This was confirmed also on spheroid assay with matrigel, where we basically put the cell into matrigel and control versus knockdown. You can see this mix, like it's not a mixed spheroid, it's a merge of the two kinetic. Then what you have is that the purple cell, the white type cell, invade immediately the matrigel, while the knockdown from FMN1 stays stuck more in the tumor sphere. is FMN1 at the end controlling these mechanoproperties of the cell? Because that was one of the main differences that we had between the 24 and the 21. And the answer is yes. We had less traction force when we remove FMN1. We had less focal adhesions, and particularly they were smaller and they were turning over faster. In some way, they were not maturing, and the vinculin staining showed that vinculin was stuck in the adhesion in the control case, but was turning over very fast in very small adhesion when you remove FMN1. And the cell stiffness at the end were also affected with softer cell when you remove FMN1. Is FMN1 at the end implicated in brain invasion? We confirm that also using this brain slice assay. Then we look at wild type versus mutant FMN1, and we observe that the cell, while following the tracks of the brain blood vessel, they were less invading when you dug down FMN1. And you have this type of tracks that you can follow. And you can also look carefully. And then when you do a 3D reconstruction, and you see that the cells really are encasing the extraluminal surface of the blood vessel specifically. Then we realize the problem of brain invasion in the absence of FMN1. And final question was, where is FMN1 located to explain, as a formin, to explain what's going on? Then we look with the actin first. And frankly speaking, it was not very clear, because what you see here is a co-expression of actin in purple and FMN1 in green. And when you play the movie, you can see that you have a spaghetti shape, more or less, for FMN1. And we had very strong stress fiber and everything for actin, which was very puzzling. Then it means that we had a formin that it doesn't mean that it doesn't polymerize actin, because indeed this formin has been shown to polymerize actin, but it's not making the bulk of what we know, meaning lamellipodia or stress fiber. And it's on a fibrous environment. And in fact, from many experiments, we show that those fibers were in fact microtubules. Then we had an actin polymerizing machinery bound to microtubule that was really making a crosstalk between those two cytoskeletons. Then in conclusions, then you can see this background essay with the grid with MOC on the left and FMN1 knockdown on the right, fixed and stained for actin in purple and microtubule in green. We have high heterogeneity in glioblastoma stem cell aspect and motility, which is linked to a high heterogeneity in glioblastoma stem cell mechanoproperties. And what we have really is a simple mimicry device to reveal the relevant 3D motility modes. With reductionist approach, we have a very good proxy that resembles the brain environment. We show that mechanoproperties are really tying the knot with invasive properties, with all the demonstration that I show you. And we have adaptive mechanoproperties of invasive glioblastoma, that are supported by this formin FMN1 expression. And this is very important for the disease also, because it means that we have to look at proteins expression, because mRNA doesn't tell you what the stroma is doing on the cell, how you have induced expression. And that's very important to have a more holistic view of cancer. Finally, what we show is that FMN1 is an enhancer of cell mechanical cohesion as an actin polymerizing machinery, but acting from the microtubule lattice. This is finally resumed in this graphical abstract. The tumor sphere or the tumor grow, encounter laminin substrate on the surface of the blood vessel. This increases FMN1 expression. You get stronger cytoskeletal cohesion between actin and microtubules. You get enhanced mechanoproperty, stiffness, adhesion, and force. And this leads the way to invasion, and we can mimic it with the grid system. Then I would just mention that we are collaborating for mathematical modeling with Nils Gauthier and Raphaël Voiturier. Then Nils talks last year about this particular paper that we had together, showing that we have stick-slip mechanism on linear motility. And I will briefly mention, and maybe Nils one time will be invited again to show that. Then we are developing another story now that is under review. And then we have used a new matrix based on honeycomb-like shape to really mimic the the triple junction that you have in blood vessel and the decision-making of the cell. And then we compare what's going on in the brain to those assays from glioblast cell, but also endothelial cell, the vascular compartment on the problem that we have of the stroma. And I will thank all the people that have participated to this story, like Michele Crestani particularly, that developed another story that I didn't have time to present today regarding screening cells with this grid assay. Pascal, that did most of the work for this FMN1 story. Tanya, that is working on membrane tension and glioblastoma, and Andrea, that works on another subject of the lab, which is the spectrin cytoskeleton. All the collaborators, for sure, and also you all for your attention. And we are seeking particularly postdocs and PhD students, and you are welcome to apply and if you are interested in what we are doing. Thank you.
All right, thank you so much. I actually had you slotted in my head for a larger. I thought you were going to talk even longer, so that's great. Actually, we have a lot of time for discussion.
I didn't know because you said to me 35 minutes, right? It's 35. I have 35 minutes. Okay, I was scared because I didn't know how the student, because I have other stuff to show you guys. But I stopped here because I was like, okay, maybe it's just 15 at the end. Then I have like all the development of Michele in particular that I would like to show.
No, this is perfect. We can get you again.
Okay,
yeah,
okay.
We'll invite you back for the other
sorry,
stories.
I was confused with the timing because one was 15, then I think, okay, maybe it's 35 for both. Then I just cut live when I was listening to the other talk. Okay, anyway, that's where it is. Thanks.
Great. So we have a couple of questions, and people, as always, feel free to raise your hand or drop more in the chat. From Metello Innocenti, says, nice story. And did you see compensation upon knockdown of the FMN1? And are other formins unregulated?
Alors, compensation, not really that we were able to highlight. There is other formin that can move, egg did, and that's fairly true. Then we can go back. I think I'm still projecting, right? Then we can go back since we have time. Then I can show you the blot back because there is a lot of interesting information, particularly here. Then if you look at the blot, for example, then you can see, for example, FOD3, which was the original one, also is regulated in the one that migrate following the tracks, for example. That's the one that is highlighted by here, and then it's shoot up by laminin. Then there is upregulation of that one. In the knockdown, we don't know if other formin light up, that we didn't look carefully at that, but definitely there is a lot other formin playing around. You have also, for example, MDA3, that is to notice, that now is on the left side only in the highly dividing one. And MDA3 has been linked to cell division and particularly furrow ingression. Then there is a pattern of formins in all those. Then you know that you have drugs also of formin that are being developed. Then this is interesting to eventually tackle more precisely the cytoskeleton without giving latranculin because you're going to kill the patient, obviously. I mean, this is, but having something that is more tuned toward those like specific cell and the specific pathway.
Awesome. The next question is from Nilet Taneja says, great work. How does laminin mechanistically increase FMN1 protein levels?
Mechanistically, then obviously it's because of an integrin. Then we have good candidates for that, alpha-6, alpha-3. That will be the first signal because it has to be attached. Laminin doesn't work really on a non-attached, just soluble. Then it means that the integrin has to bind and pull on the laminin. That's first will be the starting signal. Next, inside the cell, then the regulation of FMN1 is post expression, basically, like it's in the stability of the protein. Then potentially you have maybe a release of some ubiquitination system that go off actually when the cell is starting to attach. Then that brings up the protein level, or there is some post-transcriptional modification, or there is mRNA that is stored and not used. This is possible too. You have particularly those mRNA that I read recently a paper where you can have phase transition in the cytosol where mRNA gets stored, particularly for meiosis. It could happen also in the life of the cell, gets stored to be later used. Then you may have like this sort of mRNA in waiting mode, and then once the signal came, it's expressed and FMN1 increase. Then for the moment, we don't have like a clear explanation. We are investigating that.
All right, thank you. We have a question from Joanna Avasca. Hi, Joanna, nice to see you again, or ECU, I guess. So she says the mixed sphere assay was very cool. I agree. And is wondering what matrix was that in? And could you also talk a little bit more about how you think laminin supports the FMN1 stability, the protein stability.
Okay, then first on the matrix, then this was in matrigel. Then matrigel, this spheroid assay was in matrigel. Then why matrigel? Because matrigel is laminin rich. That's basically mostly laminin. And this is why they invade so well. If you do a collagen assay, they don't invade as much like that. I mean, they are much more constrained by the pore, and they are not very happy on the collagen. That will be for the first aspect, and this is laminin. The second aspect, then how it's regulated, then I just mentioned it before, I think. This is all this aspect of loop from the integrins and from inside the cell, and we don't have a really clue for the moment. I will mention the other aspect. We rule out an unusual suspect like yaptase, for example. We completely rule it out, but we don't know exactly what's going on. And for that, we are investigating, and we are particularly, I can show you because I'm a bit frustrated then now because of the longer talk, but I will show you I was supposed to show you, because we have a great diversity that we screen now. And we have intrapatient assay. And then now we have intrapatient assay where we have, basically, I show you an extreme slide here, just on 2D. Then we have these patients where we sorted different cell types inside the same patient this time. It's not patient outside, it's inside the patient. And then, for example, you see the clones on the left, they are non-motile. They basically like to make spheroid. While the clone on the middle have more collective-like migrations, and they like to stick to each other. And the clone on the right are like single entities. And that, regarding your question, if you do what we call then intrapatient molecular signaling now, then we have RNA sequencing analysis where you go like, basically you can sort that non-motile and motile have already different profile. And when you sequence that, the usual suspect came out. Then we have ECM receptor interaction, focal adhesion particularly. And what you can see, if you look, what moved from the motile to the non-motile are essentially alpha-6, alpha-3, which are integrins that are linked to the laminin coating. And for example, fibronectin doesn't have this profile. If you look at all those clones, the non-motile being the 2 and 3, you see alpha-6 is down in 2 and 3. While it's up in 1, 7, and 9. And alpha V, which is fibronectin, doesn't move. Then really we have a strong candidate with the integrins that recognize, and that's how the loop could go.
So since you have the perfect slide on, and I had this question because I see CD44 and I'm really biased towards it. I was also wondering, because in brain, there's so much hyaluronic acid, and how much of that you are considering sort of thinking about in future. Like how much of that affects this? Yeah.
We are currently investigating exactly. That's why you have this guy on the top right. This is basically here we confirm the RNA-seq with qPCR, and we screen for CD44. But you see already that you have a difference. It's not as striking as alpha-6 or alpha-2 integrins, but if you look and you just zoom that, many labs will be very happy with a difference like that already, because you can see that the 3 motile are about, like, I will say 3 to 5-fold higher in expression in CD44 than the non-motile. Then we have a lot to investigate. What I'm looking also is all what I have in this unknown pathway, because that's also where it's getting very interesting, is to go for the unknown, because integrins have been, meaning this is a proof of concept that the technique we are using is relevant and can really sort the system. Now, what we are looking is to eventually find alternative pathway, because, for example, integrins have been tried with selangitid in clinical trial. Then there was 600 patients enrolled, and selangitid target fibronectin. And that was a complete mistake from the basic science point of view. They were thinking about neovascularization, but in fact, what they targeted at the end is very nothing, because the glioblastoma don't care about fibronectin. What they like is laminin. Then this is where the rationale is.
Thank you so much. Next question is from Sangyun, also very interesting. I think we should talk more. It was interesting to see that NN124 cells produce large traction while being still much on a softer substrate than NN121. what would be the potential mechanism for 24 cells to generate large force with soft cytoskeletal? And then there is a second part. I can go to the second part later.
Okay. Then the large force, meaning this is true that they have the same level of force on linear substrate. Then it means that the NNI24 are really working more like as a long bipolar element, meaning that they are like building up long filaments like that, that are probably FHOD3. We have seen that in NNI24 background. To get back to the blot, just I show you back the blot, because this is the relevant here. Then what you see, what you see in the blot for FOD3, you see that this is the one that makes linear filament and focal adhesion driven. This FOD3, when you put an NNI24, the soft one, on lines, is highly expressed. Then there is an induction of FOD3. Then meaning they reinforce their long axis. Then there is long stress fiber that pull on the substrate. But it doesn't mean that the cell is stiff per se from the top point of view, meaning the cell body is relatively soft, and the cell is like a long cable of actin filament on a linear substrate. But it cannot bridge across other zones like the NNI21 does, and that has FMN1 down, because they don't rely maybe on the microtubules to help to build those bridges. And that's something that we are trying to understand, is the balance between the formin. But indeed, as you mentioned, for example, one of the molecular explanations of the two is that one rely on FOD3 and lacks linear substrate, and on linear substrate they make more force, indeed. But once they get challenged in 3D, they don't show that. And that's also why we need to have these sort of essays that are relevant, that are neither 2D nor only lines, but that bring a bit of more complexity. And that's what we are proposing.
Awesome. Sangyun also had a second part of the question asking, have you measured also the myosin contractility in those 3 different cell lines?
We have used blebistatin. That's what we have done. Like, then that was an obvious player. We didn't do any Western blot on the level of phosphorylation, for example, of myosin or stuff like that. No, we didn't look directly of myosin implications.
Okay, thank you. Thank you for the question, Sangyun. From Hani Suleiman, he's got a couple questions. So first, integrin alpha 6 binds to beta 4. So do you see an increase in integrin beta 4?
I didn't look beta 4. Let's see if we have beta 4. Then I need to look at the detail of the result. Let me see. Where is this guy? Tick, 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 tick. Then if we have, I think we didn't blot beta 4. We may have that in the bulk of the RNA-seq, but not necessarily at the level of the, let me see back. Then boom. Boom, where is it? Alpha. No, I don't know. Then we need to look at it also. No, I don't know. I don't have evidence. I need to look at this part of the mix to see if there is more detail on that. This is all new. This is a paper that we are finishing, this SP2G, which is basically the name of the method, which is spheroid SP, spreading SP2, and G like on grid. That's the idea.
Oh, interesting. Okay, cool. And then Hani's follow-up question is, do you think that there's a switch in the invading glioblastoma from integrin alpha 3 beta 1 to integrin alpha 6 beta 4?
Yeah, that's fairly possible that there is a switch indeed. And that will be an environmental driven switch in function of what they found. And the relevance of that is also the aspect of then coating, because the extracellular matrix that is around the blood vessel is mostly laminin. And the rationale is that the tumor will grow just by dividing first from the transformation that you have. Is it a neuron origin? Is it a meson? I mean, there is many possibilities, and we are not even interested in that. This is the point where the tumor has grown and then contact a blood vessel. And at the interface here, you will have this switch possibly. And that's what we are looking for with this assay. And that's really the switch will be highlighted by this approach here. Then I show you the movie of that. Then this is the tumor sphere dropping. on the grid. And on the left, you have the 11, the non-motile. In the middle, the 21 again, the mostly motile, and on the right, the NNI24. And when we drop, you can see the dramatic differences that we have. Then the molecular switch, that's where we're going to look at the basis, and we're going to look at those cells that come out first as the winner, and then we can know if there you have a very good candidate. And this is very helpful because this is very short. In 2-3 hours, you have a movie. Compared to brain slice, for example, imagine the gain of time. And what you can do, you have done, some of you maybe mitotic check, like with basically a cell on division, you can collect them by just slapping on the Petri dish. And then you can think here that you can also do that with this spheroid approach. You let the cell drop first, then you slap the dish, you remove the bulk of the spheroid, and you get attached the first cell that came out. And that have the relevant also geometry. And that's really where we're going to target all these switch in integrin, switch in molecular players.
Awesome. I'm going to add that I love the fact we are looking at more like disease progression with mechanics. That's such a cool topic to target.
Thank you. Yeah, thank you.
So there's another question from Florent asking, the NNI24 cells invade the whole brain but doesn't seem to grow. Still, they seem to be more efficient at invading while not expressing the FMN1. Could you comment on that? And do they really care about the laminin the same way the 21 cells would?
Yes, that I replied previously also by showing you back the Western blot of FOD3. And FHOD3 is really induced on NNI24 when they encounter laminin. Then the answer is yes. What you can see, they are much more collective. And there is something that is behind the collectivity also that we are tackling with the method. And that's indeed our link to the question that she asked. And then I show you this is here. Then basically what we have in this essay, this is the grid essay allow also to tackle this cell-cell junction, okay? Then basically the idea here is that there is, in the topics of glioblastoma, a cell-cell communication, particularly with nanotubes and calcium signaling that go throughout the cell. And what we are doing here is that we are using 2-photon excitation with NKH calcium. And basically we are analyzing the propagation of a calcium wave to highlight what's more collective glioblastoma type than less collective glioblastoma type in terms of behavior. And what the NNI24 emerge as is that they are the most collectively communicating, and they have a very high level, for example, of P120 catenin, connexin 43. That's where cell-cell junction. And those cells stream as collectively, then they pull on each other, and then they invade very well also. Why? Rather slowly, because they divide less. And we go back to the grow and go hypothesis, meaning you may have like clones that like more to walk and clones that are less prone to walk. And this, even if they proliferate slowly, will ultimately kill the patient. And that's a problem. And I will add another stuff since I have a bit more time, if there is time left, is that what we have highlighted also, and that's something in our intra-patient screen, is another like fabulous topic, which is linked to that. We have a mix of cells, okay? And then we have hitchhikers. And what we have, basically, we can have like those motile cells. This is a mix here. It's an early tumor sphere that has a lot of different clones. It's not purified. And then what you have here is a mix where you can see a single cell can drive a cluster of 3, 4, 5, 10 cells and drive the spheroid. And you can imagine that in the complexity of the tumor, you have also this problematic to interpret what's going on, because you may have very few motile cells, but the recurrence indeed is non-motile, is very growing, and you don't understand the problem, because in fact that was one out of 10 cells that were motile that was dragging the other cells. And that's really where we are also trying to bring information, to bring answer with our system.
Amazing. I'm going to say, though, the timing wise, you were perfect. So you don't have to worry that you
okay.
Because that gives us so much time to discuss, and that's
answer,
eventually.
Exactly. And that gives you the opportunity to show you also that. Yeah, that's good.
Right. Okay. Sorry, go ahead, Jen.
No, I think we're good. We're actually a little bit more on time than usual, so that's fantastic. So I think if people want to pop in more questions in the last minute, go ahead. Otherwise, we'll wrap up. So I just want to thank both of our speakers, Purnati and Nils. Thank you both so much for fantastic talks. And with that, I'm going to sign off of our YouTube, so we won't be recorded anymore. So everyone there, bye.